Start recording. You're recording. Yep, good. And the blue bar is coming up. Now, please come up. Yes, blue bar is there. And I'm going to share my screen. Start the broadcast, which hopefully will do something. Let me just quickly check. Yep, that should work. And we are going back. Now, can you see my screen? That's the most important thing. Is the screen visible? Yes, thank you very much. That's great. Lovely. At least that works. So that's a good thing. Oh, this is a weird, it's weird stuff. And I can only apologize. So in this case, you probably have seen I have put the Moodle and Sir Bernardo into brackets. Don't worry about the attendance. Heck knows what they are doing over in the timetabling office. So don't stress about attendance. It seems that they are under the impression that we don't need a lecture. I beg to differ, but you know, I'm not taking on the people from timetabling office. It's not a fight that you can win, and it's not worth fighting. So let's just, you know, let's not worry about it. So don't worry about the attendance. And if you know of people who you can contact, no, you don't. If you know of people you can contact and let them know that we've got a session, please do so by all means. That would be absolutely great. No, you don't want to see a fight because I'm, you know, don't worry about the attendance. Yes, actually, this is what I had planned last week with you. So all I needed to do really is pray that it works this time. And hopefully it does work. I'll tell you what, I'll just take that out quickly. So apparently there's no attendance monitoring today. But why it says that there is no lecture today, I have absolutely no idea. Weird stuff. But I looked at the comments that people made last week when my internet crapped out. I mean, Virgin Media decided to do some weird stuff in the Dover region, and that's why I was totally cut off. I couldn't even send you an email and say that I can't do anything. So I apologize for the glitch last week. Your comments actually were really interesting in the chat, and probably I should talk less and should make you do more work and make you more comment about things. Well, it was really interesting what people thought, especially when it comes to coming back to university next academic year. And I can tell you, we are currently working on a model that where we have a sort of a mixture of face-to-face sessions and online sessions. How this is going to work, I'm not entirely sure at the moment, but we are really working on it, and hopefully we come up with a good solution. Okay, so let's get started with BI301 Enzymes and Metabolism, because there are a few things that we need for the exams. What we are going to do. And so far, what we have done is we have really been quite destructive, if you think about it. We were discussing how we can break down compounds in order to gain energy. So breakdown of compounds, to gain energy. For energy purposes. And we usually used the term O-lysis, which means to break or break down. So we discussed glycogenolysis, which was the breakdown in glycogen. We discuss, so from glycogen into glucose, from glucose into other compounds like pyruvate or lactate. So we had glycolysis, again breakdown of the glucose, and then we could fuel things into the Krebs cycle, and the products of the Krebs cycle, the NADH that we produced, we could then put into the respiratory chain and the oxidative phosphorylation. We could also use lipids. I showed you how we can produce, how we can use lipids and fatty acids. So that was lipolysis. So we break down fatty acids through beta oxidation. We get lots of acetyl Coa, which we then can fuel again into the Krebs cycle and so on. So this is all the breakdown in order to get energy. What about generating things? So generating would be genesis. So we discussed how we can then, for example, make the fatty acids that we generate through lipolysis. Oh, and I forgot, of course, I forgot proteolysis, where we break down the amino acids. Forgot that. So these are all these olysis, breakdown. Genesis means we make, and we discussed how we can actually produce ketone bodies. So we had ketogenesis. We produce ketone bodies, beta hydroxybutyrate, acetoacetate, and acetone, from fatty acids, from acetyl Coa. So we produce these ketone bodies, but again, we use them to create energy. I also showed you how we can produce glucose. Can you remember what the term was for making glucose? What was that called? If we make glucose, if we produce glucose, what was that called? Absolutely right. That was gluconeogenesis. So here we produced glucose. But if you think about it, that was only as a sort of an emergency thing. We produced glucose because we needed to feed the brain and red blood cells, because they had no other ways, the brain, at least not for the first 2 days of starvation. The red blood cells don't have any other fuel source but glucose. So even if we are very low in glucose, we need to maintain this food source for red blood cells and the brain. So therefore, we had to produce glucose in this process of gluconeogenesis. So we were actually, if you think about it, we were constantly talking about metabolism where we break down compounds. But sooner or later, surely there must be a point where we run out of food stores. And if we want to survive, we really need to make sure that we replenish food stores, that we replenish what we have spent in exercise or through starvation. So what we are going to do today is we will talk about how we can replenish the medium-term fuel store, so glycogen replenishment. So that would be the process of glycogenesis. It's this one. And in the second part of this lecture, how we actually produce fatty acids. That's the long-term storage. Fatty acids are long-term. Glycogen would be medium-term storage. So let's have a look at glycogenesis. So in this process, we produce glycogen. Which organs actually store glycogen? Can you remember that? Which organs store glycogen? Yeah, liver, that's good. No, the kidney doesn't store a lot of glycogen. Yes, skeletal muscle and liver, absolutely right. So in these two organs, predominantly in these two organs, we have glycogen, so liver and skeletal muscle. I just abbreviated with SKM, so SKM stands for skeletal muscle and liver. And in both organs we have it's loud. Okay. In both organs, we store glycogen as the medium-term storage of glucose. Now, the glycogen in the skeletal muscle is exclusively for the use of the skeletal muscle. Exclusively for the skeletal muscle. So when we have the glycogen in the skeletal muscle, it is only for the skeletal muscle to be used. In the liver, however, the glycogen is used as a storage for other organs. So the liver stores the glycogen, but it does so, it doesn't use then the glycogen for itself. It's very altruistic, if you like. The liver is the storage space for the glycogen, but it will provide other organs with the glucose that is stored in the liver. The liver does not use a lot of the glucose that it gains from glycogen. So that's quite important. And there is quite a difference between the metabolism of glycogen and anabolism of glycogen in the liver and the skeletal muscle. The liver is incredibly selfless, absolutely. So the liver stores the glycogen for all the other. Okay, so, and all the lectures are recorded and are on Moodle, but not all lectures are on YouTube. That's for certain reasons. So, but all lectures are on Moodle, so you should find them there. Okay, so first of all, we need to get the glucose. So when can we use glycogenesis? Glycogenesis happens when there's loads of glucose available in the blood. We then can use, so after a decent meal, full English breakfast or something like that, we have lots of glucose in the blood, and now we can use this glucose to replenish any glycogen stores that we have reduced, diminished, for example, through exercise or overnight fasting or something like that. So that is where the glucose comes from. It comes from the food. And first of all, we need to get the glucose into the cells. And for that, transport into cells and this transport in general, if the glycogen stores are low, then we will use the glucose that is around, and in excess, we always need to maintain a certain level of glucose in the blood. So we can't go below that. But if there is an excess of glucose in the blood, we can use that to build up our glycogen stores. In the liver, we have transporters. So transporters. In the cell membrane, let me illustrate that like this. And these transporters will transport the glucose into the cell, like that. And we get the glucose immediately into the cell. Now, there is a difference between the transporters in the liver and in the skeletal muscle. In the liver, these transporters are called GLUT2 transporter. In the skeletal muscle, they are called GLUT4. The GLUT2 transporter in the liver, they have a high Km for glucose and a high Vmax. In the skeletal muscle, it's exactly the opposite. We have a low Km for glucose and a low Vmax. So what's the consequence of that? What does a high Km actually mean? Enzyme has a high Km. What does that mean? What does that tell you? A high Km. Low affinity, absolutely right. So the GLUT2 has a low affinity for glucose. Whereas a low Km means a high affinity. Absolutely right. So if we have a certain amount of glucose in the blood, cells with the GLUT4 transporter on it will very quickly pick out the glucose and transport it in. Whereas the GLUT2, in order for the GLUT2 transporter to work properly, we need really, really high concentrations of glucose in the blood. So the liver becomes only active in storing glucose when there is huge amounts of glucose there. That's the high Km. Whereas in the skeletal muscle with a low Km, they will pick up the glucose even if there is not so much around. On the other hand, in the liver with the low affinity, it can transport high amounts of glucose into the cell. Whereas in the muscle, where it has a low Vmax, it can't transport that much at a given time into the muscle. So these two transporters, GLUT2 and GLUT4, actually dictate how much glucose is transported into the liver, GLUT2, or into the skeletal muscle, GLUT4. Also, and we will discuss that in more detail, GLUT4 is a transporter that is regulated by insulin. And we will discuss that, what this means next week. Whereas GLUT2 is not regulated by insulin. Okay, so we get glucose into the cell. What is the next step so that the glucose stays in the cell? So here's our cell. The glucose is in. What happens next? What do you think? And we've discussed that for glycolysis. What happens to glucose when it is in the cell? Absolutely right. We need to phosphorylate it, and the glucose is phosphorylated in the 6 position. So we generate glucose 6-phosphate. In the liver, this happens through the enzyme glucokinase. In the liver, in the skeletal muscle, we have a set of similar enzymes which are called hexokinase. Hexokinase 1, 2, and 3. So they are different isoforms, and they phosphorylate glucose to glucose 6-phosphate. By the way, glucokinase is also called hexokinase type 4. That's what it's referred to. So glucose 6-phosphate. We have secured the glucose, so it can't go out. The transport out is not possible because there are no transporters for glucose 6-phosphate out of the cell. So the glucose has to stay in. And of course, we now could fuel it into the glycolysis, but we are already high in energy, so we don't need the energy, and we just want to put that glucose into glycogen as a storage form. The next step that is actually then catalyzed is a transfer of the phosphate group to glucose 1-phosphate. And this process is catalyzed by a mutase. And the mutase is an enzyme that catalyzes a shift, an intra molecular shift of a phosphate group. So now we've got glucose 1-phosphate. And this actually is already a good starting material in both the liver and the skeletal muscle to do something with it. So we take this, and I abbreviate it as glucose 1-phosphate. We activate it. Activation, and that happens in both liver and skeletal muscle. This glucose 1-phosphate, we let this react with, and in this case, we are not going to use ATP, we are using another triphosphate. In this case, it will be UTP, uridine triphosphate. Where did we encounter UTP? Can you remember? Where do you find UTP involved? Anyone? UTP. Where did we come across this molecule? Uridine triphosphate. Nobody? Have I lost you? Hello? Don't you want to talk to me? I feel low. No, it's not in the TCA cycle. In the TCA cycle, it was GTP. Don't think BI301, think BI300. Yes, it's in RNA. Remember, in RNA, we replace the T with a U. Absolutely right. So this is the UTP that is also used in RNA synthesis. Remember, vaguely, this is UTP that's in RNA synthesis. But we can use it as an energy source, and we can link this UTP to glucose 1-phosphate. And what we get if we catalyze this reaction, we have basically 4 phosphates here, right? So what we get is a molecule that's called UDP glucose. And this UDP is in position 1. UDP1 glucose. Plus, we cut off eventually 2 inorganic phosphates. So this UDP or UDP1 glucose, UDP1 glucose, this is the starting point for glycogenesis. This is the starting molecule that's activated glucose. It's activated in the right position, in the 1 position. And what we now can do is we can add this UDP1 glucose to an already existing chain of glycogen. So this glycogen has lots and lots of glucose molecules, and I indicate that with an N. This just simply means lots of glucose molecules. So we put these two together, and the glucose from this 1 position is then transferred to the 4 position, to 4' position of the glycogen. Is added to already existing glucose chain in the in glycogen of glucose chains in glycogen. So we chop off UDP, and this, the bracket means N. It's just we've got lots of glucose attached there. And what we get, if we do that, we attach we get glycogen N plus 1, because we added another glucose molecule to our glucose, to our glycogen. So remember, we had the glycogen that looks a little bit like that, hugely branched molecule, and so on and so forth. And what we do is we take this activated 1 glucose and add it to one of these branches. Does that make sense? And the starter molecule for that is UDP glucose or UDP1 glucose. Okay? The enzyme that we use for that is the enzyme Glycogen Synthase. And this enzyme actually catalyzes this reaction. It grabs UDP glucose and attaches it to an already existing glycogen molecule. What happens if we don't have an existing glycogen? So if we want to make a new, if we want to start a new glycogen molecule, we can't simply do that from scratch. We need a primer. We need a primer, a starter molecule. And this starter molecule in this case is a protein called glycogenin. So glycogenin is a starter protein that has glucose molecules attached. And since it has glucose molecules attached, the glycogen synthase can attach more glucose molecules from this UDP1 glucose and add more and more and more glucose. It can then, there will be branches and so on and so forth. So every single glycogen granule at its core has a protein, has 1 molecule of this glycogenin, because that was the starter originally when glycogen was produced. Okay, so we've produced glycogen and we can store it. But what happens if we have our glycogen stores are fully replenished? Now we've got lots of glycogen present, and hey, look at that, there is still some glucose floating around, more glucose than there should be. What can we do? So glycogen has been restored. Now we can utilize the remaining glucose that's hanging around and put the glucose then into long-term storage. So glucose goes into long-term storage. And what do you think this long-term storage is? Glycogen is medium-term, long-term storage, adipose tissues. Ketone bodies, not so much. Ketone bodies is, they are sort of transport forms, but absolutely right, fatty acids. Fatty acids and triglycerides, they are the long-term storage. So fatty acids. So we convert the glucose into fatty acids. This way is not a problem. For animals, the reverse process, fatty acids into glucose, cannot happen. In animals, this does not happen. In plants, yes. In animals, no. So how do we convert glucose into fatty acids? Now we know that the building blocks of fatty acids or the breakdown from fatty acids, what were the breakdown? What did we get when we broke down fatty acids? What was the product of fatty acids? What did we get in the beta oxidation? Yes, absolutely right. We got acetyl CoA. Acetyl CoA. So that was what we got from the fatty acids in the beta oxidation. And now what we can do is we can utilize the glucose. We produce acetyl CoA. And we go the reverse. So from fatty acid to acetyl CoA is beta oxidation. From acetyl CoA to fatty acid, this is called lipogenesis. And again, this means we produce fatty acids. The glucose is producing acetyl CoA through which process? How do we get from glucose to acetyl CoA? How do we get there? Absolutely right. No question marks. We produce acetyl CoA through glycolysis. And we then have the pyruvate dehydrogenase complex. Pyruvate dehydrogenase complex in the mitochondria that then produces the acetyl CoA. This acetyl CoA is, so this is the general scheme. So we have acetyl CoA. We have it in the mitochondria. We have to transport it out of mitochondria so that we get the acetyl CoA in the cytosol, or in the cytoplasm, I should say, because that is the place where we do lipogenesis. lipogenesis happens in the cytoplasm and really only in the liver. So lipogenesis, liver. Beta oxidation can happen basically in any organ that has, where the cells have mitochondria. Lipogenesis is happening in the cytoplasm of the liver. Okay, so we've got acetyl Coa. And what we can do now is we can add several acetyl Coa molecules You're not the only one, Bernardo. Don't worry about it. And don't worry about attendance monitoring. So what we can do with the acetyl Coa is we can basically do exactly the reverse operation to the beta oxidation. Actually, hang on, it's not that simple. Life is a bitch, and then you marry one. So what we do with the acetyl Coa, we actually attach this to an acyl carrier protein. This is abbreviated as ACP. And we need ATP for that. We gain back the CoA and we get acetyl to ACP, acyl carrier. So that's one thing. We also take the acetyl CoA and we carry out a reaction. Plus ATP plus carbon dioxide. This is the enzyme acetyl Coa carboxylase. And it uses, because we use carbon dioxide, there's also a vitamin attached to it. We've seen that before in gluconeogenesis. We need biotin. That's also vitamin H, or also called vitamin B7. Now, what happens if we get acetyl Coa plus carbon dioxide? Let's see if we can draw this molecule. We have, let's do it here. We have COS Coa, and then we have CH3. Now we attach a CO2 to this acetyl Coa. We get a molecule that looks like COS Coa, CH2, COO minus, or let me do it as the acid here. COO minus. So this is now a 3-carbon molecule because we attach the carbon dioxide, and this compound is called Malonul Coa. And this is going to be important for some regulation as well, as we see on Friday. So on Friday there is a lecture, I can confirm that. So Malonul Coa. And what we do with the Malonul Coa, Malonul Coa. We bind this to, again, our acyl carrier protein, like we've done with the acetyl CoA. We split off the HS-CoA and we get malonyl ACP. So, what have we got? We have got our acyl carrier protein. On one side, we have the acetyl, and on the other side, we have the malonyl. Okay? And what we do now is we transfer this acetyl group onto this malonyl group. And the enzyme that does this, the whole thing, is in a big enzyme complex, which is called the fatty acid synthase. It's a big complex. It's also called FAS. And all the reactions happen in this fatty acid synthase, and acetyl and malonyl are both attached to this carrier protein while incorporated in this fatty acid synthase complex. So how does it work? We have CoACP, CH3, that's the acetyl, plus CoACP, CH2, COO minus, that's the malonyl. And what we do now is we transfer this bit here onto here, and we chop off again the carbon dioxide. We lose the carbon dioxide, and we then get a molecule that looks like CH3CO. That's this one here, that's the acetyl, attached to CH2COACP. And this would be an acetoacetate ACP. Acetoacetate ACP. Whoa. So wait a moment. We added a carbon dioxide first, and then we removed the carbon dioxide. Isn't that a bit crazy? Well, actually, if you think about it, what we do in this process is we really make sure that we activate the right position here in this Malonul Coa. And that is why we use the carbon dioxide in the first place. And we also gain energy because carbon dioxide is a gas. And this gas has a much higher entropy that we produce here. And we know that delta G equals delta H minus T delta S. So if we increase the entropy, if this one goes up, then our delta G becomes more negative. So there is, in a way, there is madness. There is system to the madness. So now we've got this acetoacetate. What we now use is 2 molecules of NADPH. and we oxidize that, and we get H3C, CH2, CH2, CO, ACP. And we are still in this fatty acid synthase complex. Where do the two NADPH come from? Have we encountered something where NADPH is produced? Not NADH, NADPH. Have we discussed any biochemical pathway with NADPH? Anyone? Not in the urea cycle, not in the Krebs cycle. You've got one more shot. Not in the glycolysis, but you are right, Amma, it is from the pentose phosphate. Pentose phosphate pathway that produces NADPH. That is where the NADPH comes from. Okay, now we've got this beautiful molecule here. So we've got this beautiful molecule here, consisting of 4 carbons. And what we do is we add this again to Malonul Coa, Malonul ACP. Put this together, chop off the carbon dioxide from the Malonul Coa, add 2 NADPH, and we extend it by 2 carbon chains. So we get H3C, CH2, 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 C. So we've got 1, 2, 3, 4, CH2, CO, ACP. That's the next step. So now we've got 2, 4, 6 carbons. Guess what? Plus Malonul ACP. Chop off the carbon, add 2 NADPH, and we get C8 ACP. And guess what? We take this and add this to Malonul ACP. You get the gist? I don't mark attendance. Don't worry about the attendance. So we do this process several times and get finally to our C16 palmitic acid. We do this 3 times and add the palmitic acid to glycerol. C16. And we've got our triglyceride. Or we just simply send the palmitic acid from the liver to the adipose tissue, where the adipose tissues forms the triglyceride. Either way is fine. And we have stored our original glucose that we started with here. So here we started with the glucose. We got the acetyl CoA and we converted the acetyl CoA into this fatty acid into our C16 in the liver, sent to the adipose tissue. Now all the glucose that was available has been either stored in glycogen form for medium-term storage, or it has been secured in long-term storage as fatty acid in the adipose tissue. And that is basically how you store excess glucose that you don't want to go to waste. You first fill up the glycogen stores in the muscle and in the liver, and if there's something left, then the liver will use the remaining glucose to generate fatty acids through the process of lipogenesis. And that is actually how these fuel stores will be replenished. I hope this makes sense. That was the replenishment. And on Friday, we will discover and discuss how there is a fine balance between the use of glucose and the storage of glucose, because we need to finely regulate it. We don't want to employ costly, expensive, energy-consuming reactions to store glucose when at the same time we need to mobilize glucose. And on Friday, I will show you how these processes are integrated and how they interact with each other. And for that, we will look at the interplay of two absolutely essential hormones, namely insulin and glucagon. If you don't have any questions, thank you very much for listening and for participating. And I hope you enjoyed it. So, see you all on Friday for our last lecture in this series. Take care, don't do anything I wouldn't do, and enjoy your afternoon. Bye-bye.